Today I am going to do a journal discussion on this paper with the title of the non-canonical WNT receptor ROR2 is required for cartilage cell polarity and morphogenesis of the craniofacial skeleton in zebrafish. So signaling pathways, we have already seen WNT signaling pathway, which happens in two ways, either in a canonical, the usual way, or a non-canonical way. So this study is on the non-canonical WNT pathway with the receptor of ROR2. ROR2 is nothing but receptor tyrosine kinase like orphan receptor 2, which is required for the cartilage cell polarity. So as I was discussing in the seminar about this cell polarity, the spatial recognition, all those things, this cartilage cell polarity is necessary for that cranial cells to go to the cranial end and develop into a craniofacial region, and the morphogenesis of the craniofacial skeleton in zebrafish. So we are doing this study. In the zebrafish model. We do zebrafish model because many genes are closely related to the human genome and the zebrafish. So this is, as I told the title, this was published in the year 2023 in the journal Development with an impact factor of 4.6. The novelty of the study is how this ROR receptors transduce the WNT PCP signals, that is the planar cell polarity signals to median cell polarity is already known. Now this study explores the functions of the ROR receptors in the non-canonical WNT signal. So in the zebrafish skeleton development. So what they did is they collected the wild type zebrafishes and then they crossed them with the TG animals, and their progeny were co-injected at the one cell stage with gRNA. gRNA and Cas9 RNA, they are used for the CRISPR-Cas9. They are going to do a gene editing. So gRNA is a guiding RNA which guides this Cas9 enzyme to go to a particular region of the enzyme and alter it. So that is why they have used this kind of thing to mutate that wild type zebrafish. So the CRISPR-Cas9 mutagenesis is in a one cell stage embryo. So usually the embryo develops in various stages. In that one cell stage itself, they are using this CRISPR-Cas9 technology to edit, and they allow that edited embryo to grow to adulthood. They collect the sperm from the adult males and they check whether the expected mutation has happened or not, and through PCR reactions. And the F1s were intercrossed, and the F1 is the first filial generation, X is the second filial generation, and identified. And then what they did, they checked the cartilage and bone development by using some cartilage acid-free anisine blues and alizarin red stains, and they did in situ hybridization. This RNA in situ hybridization was performed with probes like SOX9, ROR12, WNT5B and GPC4, and later immunohistochemistry was done using primary and secondary antibodies, and then the image of the adult zebrafish was captured. So how did they anesthetized the adult zebrafish with a Tricaine, and then they immobilized them on a cellulose bed where they placed the zebrafish, and then they took all the pictures, and the cartilage measurements were also done using ImageA. ImageA is a software which is used to analyze all the images, and the statistical analysis were plotted. So this was the method used, and what did they found? They found that the ROR2 is required for body axis elongation and the craniofacial skeleton development, and ROR2 is required for cartilage stacking and polarities. We need that stacking and the polarity where to go and develop, that is needed. So loss of ROR2 alters the focal adhesions in the chondrocytes. Chondrocytes are the building blocks of cartilage, and the ROR2 function in cartilage stacking requires WNT binding and proline-rich proteins. These were the findings they got. So in conclusion, the data suggested that the common processes disrupted by loss of ROR2 in these contexts are the polarizing cell-to-cell intercalations that drive cartilage morphogenesis, which require the non-canonical WNT PCP pathway pathway. So WNT orthologs and ROR group form a small group in the PCP factors that regulate both cartilage morphogenesis and the CE of the axial receptor during the vertebrate gastrulation. Specifically the WNT5A in tetrapods and WNT5B in the zebrafish. And further studies are given to determine whether the non-canonical WNT path and pathways and the fat actions pathways interact during the cartilage morphogenesis. This is an observational study, so this followed the stroke guidelines.